So I'll be the third BRAF talk today, but have the tall order of following both Keith Flaherty and Neil Rosen. So I'm going to talk about a slightly different topic. I'm going to be talking about the challenge of BRAF mutant colorectal cancers and some of the problems we're encountering of resistance to MAP kinase pathway inhibition in these cancers. So BRAF mutant colorectal cancers represent 10% of all colorectal cancer cases. BRAF mutations are a well-established poor prognosis factor and predict increased mortality in stage 4 disease. And several studies have suggested that the presence of BRAF V600 mutations actually predict resistance to EGFR-directed antibodies, which are among the only true targeted agents we have for colorectal cancer right now. So obviously, taken together, BRAF mutant colorectal cancer is a clinical challenge in need of some new therapeutic approaches. And so initially, I think my colleagues and I in the field in general were excited with the advent of promising RAF inhibitors and MEK inhibitors and some of the dramatic results that you saw earlier today in melanoma. But unfortunately, while RAF inhibitors like vemurafenib were producing 60 to 80% response rates in melanoma, in colorectal cancer, in an initial expansion cohort of vemurafenib, only a 5% response rate was seen. Now, there certainly is some suggestion of some disease stabilization. There were several patients with minor responses, but clearly this is a much different looking waterfall plot than what Keith showed you earlier today. And so, a critical issue here for BRAF mutant colorectal cancer patients is what is the difference that's causing this dramatically different response. And I think there are two, to touch on some of the topics that Neil espoused on, two general explanations for what might be going on. One is we're not hitting our pharmacodynamic target. We're not fully inhibiting the MAP kinase pathway in colorectal cancer. And the other possibility, perhaps a very general possibility, is that maybe we are, but colorectal cancers are not as MAP kinase pathway dependent as their melanoma counterparts. But either way, understanding the mechanisms of resistance, both de novo resistance, but also acquired resistance is very important. I should mention on the acquired resistance topic, the median time to progression in this study was less than 4 months. So as an initial foray into this field, we actually looked at acquired resistance to MEK inhibitors using two of the most sensitive colorectal cell lines with BRAF V600 mutations. And after advancing them through progressively higher doses, we obtained resistant clones termed AR cells, or AZD6244 resistant cells, that were capable of growing freely in what would normally be a therapeutic concentration of this inhibitor. What we found, somewhat surprisingly, is that not only did these resistant clones, and here's just one example, produce dramatic resistance to the drug for which resistance was selected, the MEK inhibitor AZD6244, but also whatever change occurred in these resistant cells caused an equally dramatic shift in the sensitivity of these resistant clones to a compound PLX4720, which is an analog of vemurafenib. This was a tool compound used prior to the availability of vemurafenib itself. So after some extensive molecular characterization of these cells, we found that in both of these cell lines, actually, the same mechanism of resistance occurred. And what we found was very dramatic amplification of the BRAF gene itself. And the BRAF is shown here in red, and the centromere 7 control is shown in aqua. And we confirmed this result by quantitative PCR of genomic DNA. And what you see is in each case about a 5 to 7-fold increase in the amount of BRAF DNA, whereas in contrast, CRAF, there is no change in copy number observed. Moreover, when we looked more specifically at the differences between the parental and the AR cells, we found was that this amplification seemed to be selective for the mutant allele. And so if you look at these electropherograms, you can see that the ratio of the blue mutant peak to the red wild-type peak in each case, in each cell line pair, dramatically increases in the AR cells, indicating that there is selective amplification of the mutant allele. And this actually corroborates some mechanistic studies, which I won't show you today, but when we overexpress wild-type BRAF in parental cells, we do not get resistance, but when we overexpress the mutant allele, we do. Also interesting is when we looked back at the parental cell lines and looked very closely at our initial fish studies, we found that about 2 to 3% of the cells had pre-existing BRAF amplification, suggesting that these potentially these clones may be the founder cells of the eventual resistant clones that emerge with even higher level resistance. And also when we looked at human BRAF mutant colorectal cancer specimens, we found that in 1 of 11 cases, that about a quarter to a third of cells had some degree of BRAF amplification in this one particular case, but about 10 to 15% of the cells had very high-level amplification, similar to what we observed in the resistant models, suggesting that if this patient was treated with either a MEK or a RAF inhibitor, that this patient probably would be likely to have at least either not responded or developed very rapid resistance. And this suggests that BRAF amplification in colorectal cancer could be both a cause of acquired but perhaps also de novo resistance. So the mechanism of how BRAF amplification would actually lead to resistance to a downstream inhibitor MEK is actually quite interesting. We unfortunately, due to time limitations, don't have time to go into all of the details, so I'll try and summarize briefly what we found. So in response to either a MEK or a RAF inhibitor, we found that in resistant cells compared to parental cells, the ability of either agent to suppress ERK phosphorylation was markedly attenuated. And we did a number of mechanistic studies, and what I'll try and summarize at the bottom line, we saw that there were two main effects that influenced this. Number one is that in cells that had amplification of mutant BRAF, there was such hyperactivation of the MAP kinase pathway, specifically hyperactivation of MEK, that the basal levels of phospho-MEK and phospho-ERK were many-fold higher in resistant cells than in parental cells. So in order to reduce the absolute level of phospho-ERK to, say, the same level you would get in parental cells when you inhibit by, say, 50%, you would need a much larger percent inhibition, requiring higher concentration of inhibitor. But also what was even more interesting is we found a second phenomenon in that the level of MEK phosphorylation and activation was so high that you actually saw a change in the pharmacodynamic relationship between MEK and ERK, such that MEK was hyperactivated to the point where there seemed to be an excess of MEK for what was required for maximal ERK phosphorylation. And what we actually found is that you could inhibit active or phospho-MEK by 50 to 60% before you even started to see effects on phospho-ERK. And so I think taken as a general theme here, BRAF amplification is certainly one mechanism that can lead to hyperactivation of MEK and the MAP kinase pathway, but certainly one can envision several other mechanisms involving other RAF proteins, activation of RAS, activation of RTKs, and perhaps hyperactivation of MEK and MAP kinase signaling could be a general mechanism of resistance to both MEK and RAF inhibitors. Very interestingly, what we found, however, is if we combined RAF and MEK inhibitors, we were able to restore the ability to inhibit phospho-ERK in these cells to a similar degree as seen in the parental cells. And also very interestingly, although it's not quite as dramatic as in the resistant cells, you saw, not surprisingly, improved inhibition of phospho-ERK in the parental cells as well. And when you looked at how this translated to both cell viability and induction of apoptosis, if you look here, the dotted lines represent the resistant cells. Red MEK, blue is RAF, and the gold is the combination. What you see is that the combination of both drugs was able to shift the inhibition curve back to the curve that you see for either drug in the parental cells. but that the combination of the parental cells led to about a 5 to 8-fold leftward shift in this curve, showing that potentially not only can combined RAF-MEK inhibition potentially be important in resistant cells with hyperactivation of MAP kinase pathway and MEK, but also could be important in, or could be effective in actually improving the response in parental cells as well. And also when you look at induction of apoptosis, again, in the resistant cells, combination of both drugs was able to produce equal induction of apoptosis as seen by either drug alone in the parental cells, but the combination of both drugs produced superior apoptosis in parental cells as well. So two take-homes, again, from this slide. Number one is combined RAF-MEK inhibition could be an effective strategy to overcome resistance mechanisms where the key effect is sustained activation of ERK, but also that combined RAF-MEK inhibition can potentially improve responses de novo and perhaps can anticipate and prevent the emergence of resistant cells. So we decided to, this is a particularly important point actually when you look at why colorectal cancers may not respond as well as their melanoma counterparts to RAF inhibitors such as vemurafenib. And what we see is the majority of melanoma cells, and these are 3 cells that are quite sensitive to this inhibitor, when you go out to 48 hours of treatment, you suppress phosphoric quite readily and maintain that suppression. But in the majority of colorectal cell lines, you start to see ERK creep back. You get some very transient suppression, but by 24 and 48 hours, ERK levels are restored to 30%, 50%, sometimes even to 100% in a cell line that's not shown on this plot. And so this sustained activation of the MAP kinase pathway actually potentially may underlie the resistance of BRAF mutant colorectal cancers to single-agent RAF, or as I'll show you in the next slide, single-agent MEK inhibitor. It also suggests that perhaps whatever redundant signaling pathway or feedback pathway is operant in these cells could potentially be something that could be targeted to overcome this resistance. So if you generalize resistance to either MEK or RAF inhibitors, you find that resistant cells fall into 2 general groups, or resistant cancers fall into general groups. This is one of the few very sensitive colorectal lines we had, one of the parental cells we used in our resistance study. And you see that at 24 hours at least, ERK activity is, ERK levels are turned off by, in this case, MEK inhibitor. You also see, this is the only model, colon model that we see this in, but in this particular cell, you see that you're also able to turn off phosphoric, but these cells are obviously can survive independently of MAP kinase. But the majority of colorectal cells are like this cell line in the middle, where you do not completely shut off phosphoric with either MEK or RAF inhibitor single-agent therapy. And that's demonstrated here, is that even at 1 micromolar of each of these inhibitors, you still see some residual phospho-ERK levels. Whereas if you use a RAF-MEK inhibitor combination, not only do you completely shut off phospho-ERK, but if you look even at a very small dose of 10 nanomolar each, which really doesn't do much to basal phospho-ERK levels, you get a much better inhibition, and by 100 nanomolar each, you've completely shut the pathway off. And when you look at what happens in cell lines which do not fully suppress phospho-ERK to single-agent MEK or RAF inhibitors, you see that the combination, can induce dramatic apoptosis, whereas in the one MAP kinase independent cell line we have, not surprisingly, additional MAP kinase suppression does not lead to added benefit. So using this rationale, we actually decided to explore whether combined RAF-MEK inhibition could be a possible strategy to improve response in BRAF mutant colorectal cancer in the clinic. And so with Keith Flaherty's help, we were able to take this data to GSK, which is a company that is sponsoring the trial that he presented early results from earlier this morning. This involves both a BRAF inhibitor that's very similar to the Plexicon compound vemurafenib, as well as this MEK inhibitor. And as he mentioned, the initial responses in melanoma were quite dramatic, about an 80% response rate, with the remaining 20% or so of patients achieving stable disease. So overall, in this initial cohort presented at ASCO this past year, 100% disease control rate. So we are currently enrolling patients to a 25-patient expansion cohort, specifically for BRAF mutant colorectal patients with a V600 mutation. We are about three-quarters finished enrolling this cohort, and we anticipate we'll be able to present the findings of this trial at ASCO this spring, and we're very excited to see if this strategy does indeed improve response. We're also trying to obtain both on-treatment biopsies to answer the important pharmacodynamic question of whether we actually are obtaining complete suppression of the MAP kinase pathway in these patients, and whether that's sufficient to lead to response in many of these patients. This is the last slide I have before a summary, but one other point is that there's been a lot of work in the field of resistance to RAF and MEK inhibitors, and after Neil Rosen's talk this morning, we will have to add another resistance mechanism to this side. But again, a lot of these mechanisms can really be classified by whether they reactivate the MAP kinase pathway, whether they reactivate ERK phosphorylation, or whether they do not. And as it turns out in all of these different studies, a lot of the resistance mechanisms that lead to reactivation of the MAP kinase pathway are sensitive almost invariably to RAF-MEK inhibitor combinations. The degree to which they're sensitive, these are mostly in vitro studies, but this at least, as the field gets more and more complex, this potentially can advocate for grouping these resistance mechanisms in a functional manner rather than isolating the specific cause in each case. Alternatively, those that are independent of MAP kinase pathway are not sensitive to this RAF-MEK combination, but may be sensitive to co-inhibition of another pathway, perhaps such as P-ERK kinase. So to summarize, incomplete MAP kinase pathway inhibition, that should read, may underlie the lack of vemurafenib efficacy in BRAF mutant colorectal cancer. BRAF amplification is one potential mechanism of acquired and de novo resistance to RAF and MEK inhibitors. And combined RAF-MEK inhibition is a potential strategy to overcome MAP kinase-dependent resistance mechanisms. And a clinical trial of RAF-MEK inhibition is currently ongoing for both melanoma and for colorectal cancer. So acknowledgements, I just want to thank Bruce and the STO for inviting me to speak today, as well as my mentor, Jeff Engelman, my former co-mentor, Jeff Settleman. I was a member of both Jeff's laboratories prior to his departure for Genentech, and the other members of the Engelman laboratory. Keith Flaherty for his help translating these findings into a clinical trial. Colleagues in pathology who helped with the FISH studies on BRAF, and the Center for Molecular Therapeutics at MGH and Cyril Bennis.
Ryan, just one second before. So in the clinical trial, are you enrolling patients with KRAS wild type or?
So the trial is restricted to patients that have a BRAF V600 mutation,
Okay.
and because that tends to be mutually exclusive with KRAS, that effectively yes, but you also have to have the BRAF.
Have you seen any changes in AKT signaling in your resistant models?
In AKT signaling?
Yeah.
So actually, in these particular models, no. What we see sometimes with AKT signaling is there can be a moderate induction after treatment with either a RAF or MEK inhibitor or the combination. It's interesting actually. Some models it's a transient induction. Some models you actually see a decrease. There doesn't seem to be a real consistent effect of that. But certainly that is another possible mechanism of perhaps MAP kinase independent resistance is actually a feedback induction of P-ERK kinase, but it's not something we've seen universally in our models.
Okay, so when are we going to have these 25 patients' data available?
So we expect to we expect to present the results at ASCO this June.
We'll be there.
Just a quick question.
Yes.
So you're able to show with your acquired resistant lines that you're able to overcome that with increased compound concentration, right? So for the acquired resistant lines, you show that with higher concentrations of compound, you were able to overcome that. So in terms of just showing and answering your question about the differences you're seeing with these, the lineage difference between BRAFs and lung cancer versus colorectal, have you looked just at blocking efflux pumps or just using, is that perhaps?
So we haven't looked at efflux pumps because we actually don't, in this particular model, we don't think that was involved. What we actually found with a few manipulations that if we actually use a very subtherapeutic dose of RAF inhibitor where we were able to decrease the heightened level of phospho-MEK to the same level that was present in parental cells, we actually could shift that IC50 curve for ERK back to normal with a MEK inhibitor. So it didn't seem like it was a problem of intracellular drug delivery, if you will. It really seemed like it was a clear departure from the normal pharmacodynamic relationship between MEK and ERK.